That was a terrible quiz back in the day, so. Didn't move that one over to Canvas. Okay, so today our topic is anaerobic respiration and fermentation. And on really several of these metabolic topics, I don't need you to know extreme detail, but it's more of an overview of how does this style of energy generation work. So a lot of the pictures are similar pictures that we used in the intro to metabolism, but we get a little bit more detail here. So it'll be anaerobic respiration first, then a break, then fermentation toward the end of class. And when we start talking about anaerobic respiration, or really any type of respiration, if we're saying the word respiration in a metabolic sense, that term implies that there is ATP synthase working together with an electron transport chain. So we're going to have some kind of electron transport chain that pumps protons outside the cell. And the protons run back through ATP synthase and ATP synthase is converting ADP and inorganic phosphate into ATP. That's for aerobic respiration. That's also for anaerobic respiration. But you've got to have two parts operating in that. There's an electron transport chain, and then there is ATP synthase, both cooperating in this process. Where does the prokaryotic electron transport chain happen? In their plasma membrane, because they don't have mitochondria. They don't really have any other membrane to put electron transport chain proteins in or to pump protons across. So this is showing an example of one of those prokaryotic electron transport chains. Is this one anaerobic or aerobic? And how do you know? How do you know it's anaerobic? What do you look for to tell? Very small print up here, what you're looking for. The cycle's different. It is, the players are going to be different from what you learned about eukaryotic aerobic respiration. But what you look for is you look at the end of it, which on this picture is actually down here. Do you ever see oxygen gas involved? And it's actually right there. It's so tiny. You may not be able to see it from where you sit, but there is oxygen gas involved in the electron transport chain in this picture. Let's go back here. So right there, very subtle, we've got oxygen gas converting to water at the end. And this one is using oxygen as its final electron acceptor. So that makes this an aerobic respiration pathway. Those of you that were in cell last fall, and we had electron transport chain like going left to right, and the protons were going to the top of the page, and then we had oxygen being used at the bottom of the page. This one is turned on its side and in a different order, but that doesn't really change the essence of what's happening. So first off, this would be plasma membrane. How do I turn this off? This would be the plasma membrane. There we go. Of a prokaryote. And they're showing environment out here, like that's outside the cell, that's extracellular. So if you got this picture, you could write extracellular out here. And then here is cytoplasm. So some things I'm going to highlight about this electron transport chain. We've still got NADH and FADH2 delivering electrons into this system. So this on this slide, this end is the beginning of the electron transport chain. And the cells have been making NADH in the cytoplasm from glycolysis and Krebs cycle, probably. And the NADH is still delivering electrons into the system. FADH2 is still delivering electrons into this system. That's similar. But what's kind of different is that the proteins that pump protons by that electron energy, those are a little bit different than the eukaryotic ones. They're still named in order complex 1, 2, 3, 4, but they have a little bit different shape and identity than the eukaryotic ones. And I'll have you learn their names in here for the prokaryotes, but similar concept. So each one of these transmembrane proteins, they are passing electrons one to another, and you still have a quinone here. So you can circle this Q and say that that's a quinone, and you still have a cytochrome here, like eukaryotes do. But the proteins that go all the way through the membrane are the ones that are helping to pump protons and create proton gradient out here. So generally, same style, same deliverers of electrons, slightly different shapes of the pump proteins. This one's still aerobic respiration. So one other thing to write about this. Whether respiration processes are aerobic or anaerobic depends on whatever the final electron acceptor is. Writing this in a weird way. Whatever the final electron acceptor is determines whether this process is aerobic versus anaerobic. So remember, the purpose of oxygen at the end of aerobic respiration is to pick up electrons that have come through here, and now kind of their energy has been used up. So they've got to go somewhere. They go on the oxygen, water is produced as a byproduct. So having oxygen at the end makes this aerobic respiration, even if it's in a prokaryote. If it's not oxygen picking up those electrons, it's an anaerobic respiration style. What kind of things can make electron transport chains stop running? What kind of things can mess this up and prevent it from keeping doing its thing? What if it was you yourself? What could make your electron transport chain stop operating?
Maybe something that happens with H plus concentration that would like stop it.
Yeah, if you don't have, there's several things. If you don't have gradient, that can stop it. Like if something messes up the gradient and you don't have H plus out here, that's going to stop it because there's nothing to power ATP synthase. Yeah. So I'm going to push on that a little bit more. What could make the gradient disappear?
Keep it in like a basic solution.
A basic solution. So in bacteria, if this is their environment and they're in a very basic solution that they're not optimized for, that could make this stop. So you're depending on having proton gradient outside the cell where you've got high protons versus low protons. If the environment is super basic, soaking up a lot of extra protons, this is going to have a hard time running. Yeah, good. There are way more bacteria that can live in acid environments than can live in basic environments because of that. Here's a couple of other things that can make this stop. And I think I want to write these down. And these are things that would make the electron transport chain stop more than specifically the protons, but they're going to have the same effect in the end. If NADH and FADH2 for some reason can't deliver electrons, if they can't deliver electrons, that's going to make this stop. And most of the time, if those are not, there's two reasons why that could happen. Either the cell is starving and it never did glycolysis and Krebs cycle in the first place because it has no food source. So whatever is their fuel source is providing electrons to NADH to deliver into the system, and then they make ATP energy. So That's one reason why NADH and FADH2 may not deliver electrons would be what I'm going to call starvation, low energy supply. Or the other time when this happens is when there's a lack of the final electron acceptor. Causing you to have an electron backup in the electron transport chain. So I know I wrote that behind the computer for some of you. So option 1, starvation, and there's not enough fuel to give electrons to NADH. Option 2 is you have a lack of the final electron acceptor, is what that says. You don't have your needed compound at the end to pick up electrons that are coming out of this chain. And if there is no electron acceptor to pick them up, all of these components are basically going to be stuck holding the electrons that they've got. They can't pass it on. You've got electron transport chain full, and there is nowhere for NADH now to drop off electrons. All of this is like an assembly line thing, and to keep it going, it depends on always having a big supply of this thing, this final electron acceptor, to take those electrons away so that the transport of electrons can keep going. So aerobically, like people, if we run out of oxygen, we're going to ferment lactic acid briefly, but we can't maintain that for more than like 20 seconds, really, if we don't have an oxygen supply to keep this running. So similar, even if it's anaerobic respiration, whatever is the final electron acceptor, it's got to be there to pick up electrons to keep all this running. If it's not there, this whole thing will back up and shut down, and the organism can't do that anymore. So that concept is super important to, like, well, then what are they going to do? Or what do they need tons of? Like, we need tons of oxygen. Some microbes are going to need tons of sulfate. That's what their last electron acceptor is. So I know I've been on this picture a really long time, but there's just a lot I wanted to say about it before we go a little quicker through the next coming up slides. Okay, so that's kind of reviewing what aerobic is like and what kind of things could make it stop and what are the important players. Anaerobic respiration is similar, but there's going to be a need for large amounts of something else besides oxygen. There can be something else that's the final electron acceptor. If so, that's an anaerobic respiration style. And if so, the cell that is running anaerobic respiration has to have large amounts of the final electron acceptor, like abundant amounts of that thing. This slide is in there twice because when I made the slide set, I would write on the screen, or I would have people write on the page, and we used to write so much about this that they needed 2 pages. I'm not going to write as much about it anymore. So the same slide exactly twice, but I'm going to use one of them today. This is a redox tower with tiny font that's probably hard to read. And it's showing oxidized and reduced redox couples. So each one of these little blue arrows is the oxidized state and then the reduced state. So everything that's at the bottom of the arrow on this chart is the oxidized state, which makes it an electron acceptor. So the oxidized state, when it picks up electrons, it will become reduced. So each one of these little arrow teams are a redox couple. And they're arranged in order of the redox potential. This chart, I want you to understand the concept more than memorize any other piece of it. So technically, anything on this page can act as an electron acceptor and pick up electrons and become reduced. And each one of the oxidized substances on this page can be an important electron acceptor for some type of microbe. Likewise, a lot of times the reduced product ends up being a waste product. It was just a place to put electrons a lot of times. So down here at the bottom, we've got the oxygen-water redox couple. And this is the one that would be happening in aerobic respiration, which they say here. Oxygen picks up electrons, gets reduced to water. Water is the waste product. So if oxygen is your final electron acceptor, you need a lot of oxygen to pick up those electrons. Circle this one, or make a note about this one. But that redox couple has the most beneficial energy potential. That one has the most beneficial energy potential out of any of these redox pairs on this page. Oxygen is the best. And the way that I think about that concept like, I would never say this in front of Dr. P. He probably wouldn't like mark me, but he might give me a look. Oxygen can pick up the most worn out, low energy electrons because it's so electronegative. Oxygen gas is a great oxidizer. It will take almost, super low energy electrons. It'll still take them when they have intrinsically low energy. So over evolutionary time, the ability to pass electrons onto oxygen essentially lets the organism wring more energy or gather more energy out of the electrons and like have a final heave of worn out electrons onto oxygen and not these others. So if an organism can run aerobic respiration and put electrons onto oxygen, that's going to end up making it more ATP. So we can put a therefore on here. Therefore, doing aerobic respiration essentially unlocks more energy and leads to greater ATP production. Oxygen is the best. So once aerobic respiration developed, way back when in prokaryotes, the organisms that could do that are making more ATP. And they've got more energy than their neighbors. And they kind of exploded as a group, as a really successful group, because they're making a little bit more ATP every second of the day. And then that greater amount of ATP later on allowed for more complex activities to happen in microbes and then eventually in eukaryotes that run aerobic. Aerobic respiration will always give the most ATP. And if an organism can do that, it's going to do that. Like we don't do fermentation to make lactic acid in our bodies unless we've got an oxygen deficit, because it's lame. It doesn't make much ATP. So what the other implication of that is that any of these other types of electron acceptors, the higher up you go on this list, the wimpier the ATP production is from that style of anaerobic. So this one's aerobic. Everything from here up could be used in either anaerobic respiration or as like an intermediate step. But say up here, if you're running a metabolic style and you're using protons as your electron donor, or sorry, as your electron acceptor, and then producing hydrogen gas, like that's kind of a barely low energy style metabolism. So kind of the further up you go, you get more primitive and less energy production. This one is the oldest and the wimpiest in terms of ATP production. If you're just producing hydrogen gas as your waste product, that's just kind of barely getting by energetically. Last thing I'll mention about this before the break is that the ones up here that are kind of wimpy are mostly done by the obligate anaerobes that can't even deal with oxygen anyway. Most of these redox intermediates are being used by the obligate anaerobes. They're going to die in the presence of oxygen, and that style of metabolism is older and ancient because it developed in the days before oxygen. Let's see. The last thing I want you to do on this is to somehow highlight, circle the nitrates. So there's one right here, NO3 minus, NO3 minus, and the sulfate right here, because we'll be talking about those a little bit later. Nitrates are kind of energetically pretty good as a final electron acceptor. Sulfate's kind of medium. Protons are the worst. But if that's what you can do, that's what you can do. And then just a reminder, if an organism can do anaerobic or aerobic, if they're facultative, like this statement is about facultative anaerobes. If you've got the option to do aerobic or anaerobic, and you are in an oxygen environment, aerobic is always the choice because you're going to make more ATP. Most microbes that are medically important, if they're doing anaerobic respiration, they're using nitrate or sulfate or CO2 as their final electron acceptor. So these would be kind of like the top 3 alternative electron acceptors that are most common for the medically important anaerobes. And that's where I'm going to end on anaerobic respiration. Let's have our break, start back at 9, talking about fermentation next. Okay, so the rest of today, I'm going to talk about fermentation. And before I had microbiology, fermentation was basically always about if you're exercising and you're in an oxygen deficit or you're sprinting or so on, you're going to make lactic acid, makes your muscles hurt, and then that was kind of it. So doing fermentation kind of makes more sense when you relate it to what microbes are doing. So to start talking about fermentation, it's important to remember what's going on in glycolysis. Because most organisms, no matter how ancient, they're going to do some kind of glycolysis to start off their metabolism. And usually that's based on a 6-carbon sugar, a hexose. For eukaryotes, we prefer glucose to be that sugar most of the time. But during glycolysis, you've got some ATP input, but greater ATP output. So you're making 2 ATP net, a little bit. And then the other good thing that you get out of glycolysis is you're putting some energized electrons onto NADH. So ATP is good. That's usable by the cell as energy. But NADH with energized electrons is also good because those can go to electron transport chain and help power its activities. That's generally what's going on in glycolysis. So to summarize, glycolysis makes a little ATP and some electrons for electron transport chain use. It's a start at energy metabolism. So the problem comes if something happens with electron transport chain where you don't have the final acceptor around. This is getting back to when I said what could make electron transport chain stop. If you don't have the final electron acceptor like oxygen for us or nitrate for microbes, all the electrons get backed up in the electron transport chain. Not that many pictures away. You don't have the final electron acceptor. All of these are stuck holding the electrons that they've got, and NADH cannot dump electrons into this system. NADH and FADH2 are going to be all reduced, and there's no NAD plus or FADH to pick up new electrons out of glycolysis. So if electron transport chain stops, and NADH now is all going to be reduced and not recycled back to NAD plus, eventually glycolysis will also stop. All of this depends on a constant handoff of electrons, one to another, starting with NAD plus, picking up electrons in glycolysis. So electron transport chain backs up. NADH has no place to put electrons. The cell is going to use up all of its NAD plus supply pretty quickly and then have nowhere to put electrons. And that's really bad because in that case, you're not even making the puny 2 ATP per sugar in glycolysis. That would be really bad. So what fermentation really is, fermentation is making a place to put electrons get those electrons off of NADH so that you rebuild NAD plus and you at least keep glycolysis going and make a little bit of ATP. Fermentation is never the best option, but it's a useful backup option. Backup option. That's what fermentation is. Find a place to put those electrons and keep glycolysis going. So let's compare that to this picture. You've got a pretend bacterium here, that's what this represents. And it's got to get fuel, some kind of energy, food source, from the environment, and it will bring it into the cytoplasm, and it will start the process of doing glycolysis with that compound in some way. So this could be mannitol. Some microbes could use mannitol to do this. It could be glucose, it could be lactose, and they're breaking it down, putting some electrons onto NADH, getting a compound that they can do substrate level phosphorylation and make a little bit of ATP. This is the little bit of ATP, like from glycolysis. And then they end up with an oxidized compound. So what fermentation is doing is basically NADH, if it can't drop off electrons at electron transport chain, it's going to put electrons onto this thing. This becomes a place to put electrons, then producing a fermentation product. The fermentation product is a waste for the cell. So you could write here next to the box, fermentation product, waste. And from the microbes or our standpoint, the waste product is just a place to put electrons so that glycolysis can keep going. And the waste product eventually needs to get out of the cell in some way. the place to put electrons, I'm just going to add into this board writing over here. That's the waste product. What are the major wastes produced during fermentation? Or here's a more obvious way to ask that. What do we get out of microbial fermentation that's useful to us as people? What's your favorite microbial fermentation product? What's the major fermentation product in humans? When we ferment, what do we make as our waste? What's your favorite fermented feed? Salt. Sometimes we need salt to create fermented food, and fermented foods tend to be salty.
Kimchi.
Kimchi. Yeah, kimchi is a fermented food, and it's salty. What else does it have in it? What's the real fermentation product? It's the starting product. So the starting product would be the sugars coming out of the cabbage or whatever. And then the microbes are acting on that in a salty environment. And then they're making acid a lot of the times. So your three major fermentation products of microbes are the following. So write this here. There are acids. There are a lot of times alcohols. And a lot of times CO2 comes along with that. CO2 is a lot of times not the direct fermentation product, but it tends to be produced along with those. So when you make bread and put the yeast in, or sourdough starter, there's a fermentation process going on, and the CO2 that comes out of that process rises or raises the bread. Or alcohol production depends on microbes fermenting alcohol and producing it. From their standpoint, the alcohol is just kind of like an accident. They're not making it for themselves. We're finagling them, so to speak, to get them to make alcohol. Or when you make yogurt, cheese, microbes are fermenting lactose and producing acid as the fermentation product, and the acid makes the cheese solid, or it makes the yogurt thick. So taking advantage of fermentation, humans have been doing that for years, but a lot of times the product is some kind of acid, alcohol, and you usually get CO2 coming along with it. Those are the major products. So when we talk about fermentation, the way we describe it can be two different ways, and this can be confusing. Fermentation can be described either by what's being fermented. So way number one, you can describe it as what's being used as starting material. What's being used as starting material. So an example of that would be mannitol fermentation. Like the organism can use mannitol as an energy source and then produce acid as a byproduct, and the acid changes the color of the pH indicator. Or lactose fermentation, that's named by the starting material. And that way is named after the organic compound that's starting the whole process. So the starting material, that's the organic compound. We can talk about that style of fermentation. The other way of talking about fermentation is to name it after the waste product. So style number two, did I write that? Is to name fermentation by what's being produced as the waste product. So when, if we talk about microbes fermenting alcohol, or when we look at in a few minutes something called mixed acid fermentation, or lactic acid fermentation, those are talking about what the waste product is. And there's not, I don't know, I don't love these naming systems because it's not always obvious which type the person is talking about. So if you ever have a question about which type I'm talking about, just ask me. But you know, breweries, they care a lot about alcohol fermentation. Alcohol fermentation is not using alcohol, it's producing alcohol as a waste. So when people talk about fermentation, they might be referring to what's the starting material, or they might be referring to what's the waste material. And for the purposes of this class, if you're not sure, just ask me and I'll clarify. This chart is talking about common bacterial fermentations, is referring to what is the waste product. So if you look here and it says alcoholic fermentation starts with a hexose and it produces ethanol and CO2. Something that's so cool for me to think about is that like every carb source of every society, they found a way to make alcohol out of it. So like sake in Japan, rice. What's the carb source for vodka? Potato. What about tequila? What do they start with? The weird one. Agave. Yeah. Beer. It uses yeast. Wheat. Sometimes you add in hops so you get a little bit different taste. Like you've got your corn, moonshine. Every society starts off with some kind of carb that they get hexose out of, and has developed over the years alcoholic fermentation for their culture. Some of you that go into like research, or if you go into industry, a lot of our industrial acids are made by micro fermentation instead of producing them through chemical synthesis. So butyrate and butyric acid, when you buy those as chemicals, they're usually produced by microbes as a waste product. So any of this stuff, like acetate, can be produced by microbial fermentation, and a lot of times that's cheaper to make these in fermentation vats than to make them through chemical synthesis. And then this chart is more focused on strategy number 1, what's being fermented. So that previous one, you might write the number 2 on it. Those are named after the waste products. These kind of fermentations are listing what can the starting material be that the microbe uses at the beginning of fermentation. So as a group, microbes can break down acetylene gas, that's pretty cool. Citrate, like citric acid cycle, sometimes that can be the starting material. Benzoate, and so on. Make a note here or on the previous. Just about this. Specific microbes do specific types of fermentation. Not all microbes can do every kind of fermentation in the world, just like not all microbes can break down mannitol and use it as a fermentation starter. Or not all microbes can break down lactose and use it as a fermentation starter. It depends on the specific type of microbe. These 2 slides are completely out of place. So we're not going to talk about that today. Talk about that later. And I want to just stay on this for a minute and end here. In the lab, when we are looking for, like checking microbes for what they can ferment and what they produce as a waste product, anything where we're looking for, does it make acid? And then it changes the pH indicator. Like that's checking some kind of fermentation capability of that microbe. Like McConkey agar. You ferment lactose, it's making some acid byproducts, and then that's reacting with the agar and the colonies, and they turn opaque. A lot of times we're looking for the acid to see, can this microbe turn this thing into this waste product? And a couple of ones that I want to just highlight what their definitions are homolactic and heterolactic, just to get those definitions. So if we say that a microbe does homolactic fermentation of whatever, what that means is that its waste products are essentially lactic acid. So just lactic acid is the waste product. If we say that an organism is doing heterolactic, I want to write the whole thing again. So if we say that something is doing heterolactic fermentation, the waste products are lactic acid and something else alongside it. The names kind of make sense, like Homolactic is lactic and lactic, all one thing. Heterolactic is, it makes lactic acid and it makes something else. So sometimes, a lot of times the something else is ethanol. So usually ethanol. And that's the one that breweries said it usually care about the most is ethanol. And then one more definition, organisms that do mixed acid fermentation produce wastes that are more than one different acid. So they're making multiple acids as a waste product. That's mixed acid fermentation. Mixed acid fermentation. So they've got an example here that there's some organisms that can break down a 6-carbon sugar and make ethanol and butanediol and succinic acid, lactic acid, acetic acid, and formic acid. All of it. Mixed acid fermentation. Generally, the more types of waste products that a microbe can produce, like the more it's going to stink, honestly, because some of these byproducts will have a smell to them, and the gram-negatives as a group are doing more types of fermentation than the gram-positives, partly because they come out of our gut, and they've evolved to use all kinds of starting materials and make all kinds of wastes. And they're doing a variety of metabolism, and they're making a variety of stinky wastes. Is why they smell that way. That's the end for today. Thursday is going to be about nitrogen fixation, and quite a lot about plants and soil. So that's coming up Thursday. Don't forget to at least start a plate for your unknowns today. And don't forget to write your initials on the number 2. That's how you know that it's yours. So, see you Thursday.